Perfect.
So first of all, let me thank the organizers for inviting me to give a talk here today. I will be talking about several of the computational resources for IDPs we have been developing in the lab over the last couple of years. First of all, a few words about IDPs in general. You've already heard about intrinsically assorted proteins and regions. This is just a brief recap. So in reality, we're talking about a variety of situations, which goes all the way from things which are disordered and dynamic, shown here as being highly flexible, to disordered and compact, where there is not so much variation. At the other end of the spectrum, we have things which are disordered and extended, and others which still have a residual structure. And shown in blue here are binding segments, because disordered proteins themselves are obviously only the scaffolds on which a number of binding sites reside. Two of them are shown down here, where you have the crystal structure of their binding partner in gray and the small peptide in light blue, which corresponds to the part being disordered and folding upon binding. So the general idea for the human proteome is that out of about 21,000 human proteins, encoding about 11 million residues, 3 to 4 million residues are intrinsically disordered. So it's a large fraction, it's over a third. And one of the key advantages for the intrinsically disordered binding regions in particular is that they offer more accessible surface per residue. So it is easier to, in a way, to get more area on which to get the peptide to bind. And this is important because it allows more flexibility, of course. So this is just a very brief overview. The first tool I wanted to show you is trying to summarize several of the features for any protein of interest. And in fact, it's a fast predictor. It's called FELS. And the acronym stands for Fast Estimator of Local Latent Structure. And the goal here was to have a tool which can be applied to any sequence of interest and returns results within a few seconds by giving all the different propensities for secondary structure, disorder, the presence of aggregating regions, hydrophobic clusters, and the presence of charged clusters, or even amphipaticity, or the composition of the sequence. So let me show you an example and how this can be useful. The example would be to P27, which is the cyclin-dependent kinase inhibitor. So this is a fully disordered protein of around 150 residues, which binds to the CDK cyclin complex. So there is a crystal structure which is shown here in gray, and the part of P27 which binds is shown here on the side. And the kind of output you get from FELS is these histograms here, where you have a zoom in of the region shown here, which is the one binding to the CDK cyclin complex. And you have various features. Remember that these are all predicted on the fly from the sequence. So you can also add any other construct which may be of interest experimentally. And you can get the propensities for alpha helix here and beta strand in these two different colors, the presence of disordered regions, and then amphipaticity. And you can see here that this amphipatic region corresponds very well to the helix, which is shown here on the structure. So by having this type of analysis, you can get a feeling for the things which could be binding upon, folding upon binding, and the ones which are more disordered and which will not be able to do this kind of binding. So it's a tool which can be of use for some of the proteins of interest. With this being said, I wanted to turn my attention to several of the databases we have been developing over the years. The first one, which is probably also the best known one, is DisProt, which was initially published in 2005. And this was for a long time run by Keith Dunker's group in the US, until it was brought to Europe and moved to a community curation platform in 2017 with DisProt 7. And it's now hosted by my group here at the University of Padua. In total, here you see some of the faces of the people involved. There are over 60 people who have been contributing to the latest release of the database. It has been considerably improved over the years and now has over 2,000 manually curated proteins which have been annotated. We also recently published this current protocols in bioinformatics, which is a tutorial on how to use the database, and it's widely cross-linked to other resources. So what DisProt actually annotates, and this is typical for the sort of basic annotation you can do for IDPs, is that you have these feature viewers, as is shown here, where you have parts of the sequence here, in this case, the N terminus and the C terminus for P53, which have been annotated as disordered. And each of these segments here is backed up by annotation of the region, which has its own ontology. So we have a disorder ontology by now, where it says here, for instance, that this is a cis-regulatory element, and tells you the fragment and the method on how this was determined. But mainly the information in DisProt is contained in these information fragments as shown here. So for each of the regions being annotated as disordered, there is information about the ontology term, for instance, disorder, the fragment, the curator who did this annotation, the experimental method used to determine it, the statement, if it was available in the original publication, if there are any cross-references, and above all, the scientific publication to back that statement up. So as I was saying, DisProt is manually curated data, so it's comparatively few. It's about 2,000 proteins, but you can rest relatively sure that the information is extracted all from the literature and is correctly interpreted, so that there are experiments backing up those statements. And this is one of the key points for IDPs, because there is always relatively scarce evidence which you have to collect throughout the literature to know that something is truly intrinsically disordered. And this is one of the databases. The second one I wanted to briefly introduce is called MobiDB. And this is a different attempt to analyze proteins for intrinsic disorder. It is based on the idea that there are different sources and definitions of intrinsic disorder. At the top of this quality pyramid, you would have curated data such as DisProt, but below that you can have indirect sources. We've heard already about NMR and X-ray crystallography being used to infer parts of the proteins as being intrinsically disordered. So this information can be leveraged. I will not go into the whole discussion of what is the true disorder and what only has features which are similar to a disorder, because that can be a lengthy discussion. And in this sense, MobiDB is agnostic to the various definitions. It just presents the various information one alongside the other. And these indirect sources, you can think of them as being available for over 30,000 proteins nowadays, mostly from the protein data bank. And below that, you can have predictions, which of course have the lowest quality because these have to be validated, but you can run predictions if you have fast predictors for all of the known protein sequences. And this is what the bottom layer of this pyramid says. So in fact, we now have predictions for all known UniProt sequences. MobiDB, I should also say, this was first published in 2012 and was the work of a brilliant PhD student at that time, was Tomás de Domenico. And we have continued to develop it and expand it. The latest version was just published in Nucleic Acids Research a couple of days ago. And by now it has become quite a complex setup, because MobiDB integrates a lot of different sources of information. So we have various member databases as shown here. And several of these are databases which curate intrinsic disorders. So of course, DisProt. We have IDEAL, which is another database hosted in Japan. We have some other specialist databases which have emerged over the last couple of years, like DIBS, MFIB, FastDB. ELM is the eukaryotic linear motifs database, which is for short linear motifs. So it's not exactly the same thing, but it's still related to disordered. And of course, UniProt KB also has some regions which are intrinsically disordered. These are the sources of curated information. Then we have several primary databases which we use to extract additional information. For instance, the protein data bank is used to derive information about missing residues and mobile residues, the mobile residues being the ones with the higher B factor, for instance. And then we also use the protein data bank to infer linear interacting peptides. So these are our proxy to determine the folding upon binding regions. And we have several additional modes of information. At the very bottom of this, we use all the UniProt KB sequences on which we run several of the predictors. There are various layers of quality. This is the current data quality pyramid for MobiDB. So it has been expanded over the years to include also information about homologous sequences. So say, for instance, DisProt contains information about P53 in human, which has an experimental backing, but it doesn't contain information about P53 in mouse. And this would be then annotated via homology. The database is available in various forms, mostly through the MobiDB website, which is human readable, but it also contains an API which makes it machine readable, and therefore we have a couple of power users who have been crunching data on several million sequences, and this is all possible thanks to the current setup. MobiDB data is now also available through Interpro, UniProt KB, and the PDB Europe, thanks to collaborations made possible through Elixir. And it currently hosts predictions for over 189 million protein sequences. But let me give you an example of the type of output you would see in the simple view for now for P53 in human. So we have some information, some basic information about the protein, what it looks like. Here are the interactors, which are also pulled from the database. So by clicking on any of those, you would go to the corresponding entry page from MobiDB, and this can be useful to analyze the surroundings of your protein. And this is the simple view where we have a feature viewer annotating for the disorder consensus. This is mostly done by MobiDB Lite, which is a consensus predictor. We also have subregions annotated for different flavors of disorder, which have been identified. And then we have a consensus for the linear interacting peptides. So if there is evidence about this region in structure being an interactor of another protein and likely undergoing folding upon binding. This is just the simple view. There is a more advanced view, which is an alternative to look at the data for those who are interested in drilling much deeper into the data itself. And here you can see that, for instance, we have about a dozen different predictors for intrinsic disorder, which are shown here. So you can see really all the gory details about which predictor tells you which residue being disordered. We also have information about predicted secondary structure over here. And the information about the linear interacting peptides is connected to the protein data bank. So you can really go back and see which crystal structure or NMR structure the information came from. So this can be explored much further. We also annotate the regions for post-translational modifications, and phosphorylation sites in particular can be relevant for disorder to order transitions. And here on the right-hand side, you see another feature of MobiDB, the advanced view, which allows you to see for the various structures of complexes from the protein data bank, the interacting region. And here you see highlighted the amino acids which are interacting. So here you can more easily see which are the interactions defining the interaction between this peptide and its corresponding binding partner. So finally, I would just briefly like to mention another database which we have recently renovated, which is PED, the Protein Ensemble database. And this takes a different view. Here, we're really looking at the structural ensembles of which have been experimentally determined via NMR, SAX, or FRET. It is a repository for these structural ensembles, and it is manually curated. It has quantitative and qualitative descriptors available. At present, there are over 100 different proteins for which this information is collected, and this is a collaborative effort, including a dozen different experimental groups who have been contributing to the database. You may want to have a look at this if you're interested in the actual ensembles. Finally, I would like to acknowledge the people in my lab who have been contributing to this effort. There are several people involved in maintaining the resources. One in particular I would like to highlight is Dr. Damiano Piovesan, who is the project lead for our various IDP databases. Our main source of funding is the European Union through a Marie Curie RISE consortium called IDP Fund. And we also would like to acknowledge support from Elixir, through Elixir Italy mainly, which is for the infrastructural part. And just finally, for those who may be interested in hearing more about these type of computational resources, the IDP Fund Consortium runs a series of seminars, and the next webinar is scheduled for the 22nd of January. If you're interested, you can register for the webinar at the URL down here. And with that, I would like to thank you for your attention.